the accessibility of regulatory factors to DNA in mammalian cells. DNA contains our genetic information as a sequence of nucleotides, adenine, thymine, cytosine, and guanine. And in our nucleus, DNA is packaged as chromatin. And chromatin has multiple different functions, including maintaining proper DNA replication. So as you go through the cell cycle, the DNA is replicated and distributed between two daughter cells, and chromatin maintains accessibility of regulatory factors to these processes. In addition, during normal and external mechanisms, DNA damage can occur, and chromatin also contains or controls this accessibility to DNA damage repair. However, this thesis will specifically focus on the functionality of chromatin related to gene transcription. And we know that—
I'm so sorry. I promise I will not interrupt again. One quick suggestion. You want to shrink the bar with the
sure.
people on the
Yes.
right, because otherwise it covers some of the graphics.
Okay.
That's perfect. Thank you.
Okay.
Sorry.
Sorry about that. Okay, thank you for that. Okay, so we know that gene transcription occurs when RNA polymerase and transcription factors bind to the promoter of genes that are being transcribed, and transcribes DNA into RNA, which is then spliced and exported into the nucleus to be translated into proteins. And the entire transcriptome and proteome of our cells constitutes what we call cell phenotype, which defines the functionality of cells. So each of the cells in our body, this includes stem cells, intestinal cells, red blood cells, have the exact same genetic information, but a different transcriptome and proteome, partially due to the regulation by chromatin itself. And this means basically that chromatin can influence not just normal differentiation, but also contribute to a malignant state. And we know that chromatin structure is interrelated with transcription, for example, in diseases such as cancer. At the level of the genetic code, certain genes and the nucleotides can be mutated to influence the functionality of proteins. Above this level, chromatin is organized at the level of nucleosomes, which are composed of DNA that's wrapped around histone octamers. And each of these octamers have intrinsically disordered histone tails, which can be chemically modified to form our epigenome. And the epigenome contributes to local accessibility to transcription factors. So a more heterochromatic epigenome contributes to a more closed, less transcriptionally active gene, and a more permissive or open euchromatic epigenome contributes to increased transcription because the genetic region is accessible. And for example, in cancer, different marks such as increased methylation can influence the overall differentiation of cells and their overall plasticity in response to certain stressors. Now, above the level of the epigenome, we have supranucleosomal chromatin structure, which is on the order of hundreds of kilobases to megabases. And basically, this can contribute to long-range four-dimensional contacts. And what I mean by four-dimensional is in 3 dimensions as well as over time. We know that transcription is regulated when specific genes with their promoters come in contact with enhancers, which you can see here. And these are located a large distance apart on the linear genome, but the supranucleosomal chromatin structure, such as this CTCF cohesin loop that you can see here, contributes to these long-range four-dimensional contacts. And for example, when certain supranucleosomal structures are disrupted, we can increase the gene expression or decrease it of oncogenes or other types of genes that contribute to the cancer state. Now, altogether, we have 6 gigabase pairs of genetic information that's packaged as chromatin into just a 6-micron nucleus, and we know at these many different length scales, the chromatin contributes to cell phenotype and disease states such as cancer. However, and this thesis will focus on specifically what exactly is the supranucleosomal chromatin structure above the level of the nucleosome organization, but much below the level of chromosomes compacted into the nucleus, and how does this contribute to transcription? So a previous textbook view of chromatin is that it's organized into these beads on a string fiber composed of individual nucleosomes. And this is organized into a highly ordered 30 nanometer fiber, which then packs in further to form interphase chromosomes in the nucleus. However, recent experiments have disproved the existence of this 30 nanometer fiber, really begging the question, what is the supernucleosomal chromatin structure? And there are several different techniques that can be used in order to define this. And these include biochemical sequencing-based techniques. Recent advances in high-throughput sequencing have given rise to the advent of high-throughput sequencing chromosome conformation capture, which is basically able to measure chromatin-chromatin contacts throughout the entire genome. And this specific technique, you can see, really gives out this two-dimensional contact map of the genome topology, and this has allowed us to identify structures called topologically associating domains, which you can see are increased red regions here, which determine an increased self-self contacts of chromatin within these domains. However, these techniques are mostly bulk techniques capturing the average behavior of millions of cells, and additionally, they only give two-dimensional information at one point in time. So microscopy techniques can be paired with biochemistry techniques in order to better understand this organization. So optical microscopy and recent developments in super-resolution microscopy have allowed us to probe the chromatin structure down to about 20 nanometers. And labeling the topologically associating domains identified with these chromosome conformation capture techniques, we have actually seen their existence in individual nuclei, and you see that heterochromatic domains form these compacted nanocompartments. In addition, besides optical microscopy, we have electron microscopy, which can probe the chromatin structure down to even finer detail. And such electron microscopy studies have actually discovered that chromatin doesn't organize into the very structured 30 nanometer fiber, but actually is a disordered chain with diameters ranging between 5 and 24 nanometers. However, the existence of structures such as topologically associating domains don't always dictate functionality. For example, when previous studies have mechanistically perturbed the structure of topologically associating domains, changes in gene transcription aren't necessarily observed to be significant. So this kind of begs the question, what exactly are fundamental functional units of supernucleosomal chromatin structure at the level of individual nuclei, and what mechanisms influence the structure and function of these supernucleosomal domains? And in addition, how are their structure and function each modulated by a realistic nuclear environment? And what I mean by a realistic nuclear environment is two different components that we take into consideration. One is that the eukaryotic nucleus is a dense, non-dilute environment. And what I mean by that is within the nucleus, chromatin density, which is labeled here by this chromatin electron microscopy technique, is very dense and heterogeneous. So we can't consider each or transcription, for example, to occur in non-dilute conditions. In addition, we know that chromatin is a negatively charged system. Chromatin is composed of DNA, which has a highly negative charge on its phosphate part of its backbone, and when DNA wraps around histone proteins, the positively charged basic residues of these histone proteins, as you can see in blue, only partially neutralize the negative charge of DNA. So we need external factors in order for the 2 meters of DNA to pack into the 6-micron nucleus, as we see in human cells. And this can include physical, chemical, environmental conditions, such as ions, and these are necessary to mitigate electrostatic repulsions. So the guiding question that really underlies this thesis work that I performed is, how does a realistic nuclear environment impact the chromatin structure and function? And today I am going to outline the three major chapters of my thesis. And first, I will define fundamental supernucleosomal packing domains. Next, I will investigate the functionality of these domains and how their organization modulates phenotypic plasticity, specifically in cancer cells. And then finally, I will talk about work where we are modeling how the physicochemical environment is influencing both the structure and charge of chromatin. So starting out with identification of fundamental supernucleosomal domains, in our group, in Igal and Vadim's group, we not only consider these two types of techniques to be a very important lens to investigate chromatin through, but we also add polymer physics, which really allows us to uncover principal mechanisms underlying chromatin structure and function. And what I mean is that we analyze chromatin from a polymer physics perspective. So given this polymer right here, you can see it's composed of individual monomeric beads. And we can also think of chromatin as a polymer where each of these monomers can be composed of a unit such as a base pair or a nucleosome. And one concept in polymer physics is this power law scaling relationship between the number of monomers, or the mass of the polymer, and the three-dimensional physical space it occupies. And the power law scaling coefficient describing this relationship can be called the chromatin packing scaling. And what I mean by this specifically is that when we start, for example, from the center of this polymer and then count the number of beads in spheres of increasing radii, this defines the scaling relationship, again, between the number of monomers and the physical space. And in polymer physics, we have these simple models that are able to describe these scaling relationships for different systems. So we have, when monomer-solvent interactions are favored, this swollen coil self-avoiding walk, and then this random walk, and a collapsed fractal globule, which is a specific example of D equals 3, when monomer-monomer interactions are preferred. However, these scaling relations are only defined for these simple systems which occur in non-dilute conditions, and in addition, these systems are homopolymers, which means that all of the monomers exact the exact same chemical behavior. However, in the nucleus, chromatin is complex because it has different chemical behavior for each of its nucleosomes. For example, the epigenome or protein binding is very heterogeneous throughout the nucleus. And in addition, the nucleus is a very dense system. So this gives the question, what is the scaling behavior of the complex chromatin polymer in the nucleus? And in order to answer this question, we have a collaboration that's ongoing with Professor Vinayak Dravid and students in both Vadim Bachman's and Dravid's group, including Yue Li, Vasundhara Agarwal, and Wingshen Li, have performed experiments where they specifically label chromatin with a click-EM technique that was developed by the O'Shea and Ellisman group, and then image the chromatin density distribution using scanning transmission electron microscopy. So this allows us to get a three-dimensional reconstruction of chromatin density down to less than 6 nanometers of resolution. So this is almost down to the level of a DNA base pair and much below the resolution of traditional super-resolution techniques. So performing this packing scaling analysis on chrome stem tomograms, we are able to distinguish these packing domains, each of which have the same statistical packing behavior within them. However, the supernucleosomal organization, as you can see here, denoted by different densities in green and purple, is actually distinct between these domains. So as a next step, we wanted to observe the overall distribution of structural properties of these packing domains. And in order to do that, we take this tomogram, which gives us the chromatin density distribution. We then perform analysis from the center of the domains and identify a packing domain as when it deviates from a certain range of scaling behavior for this mass scaling curve. And we see that these packing domains have a very heterogeneous distribution of the chromatin volume concentration, which is the average chromatin density within the domain size, as well as the domain packing scaling. So now that we have seen that, in fact, there does exist a regime of power law scaling within the nucleus, that these packing domains that have been identified are heterogeneously distributed in terms of their structure, we also wanted to figure out if there are fundamental organizational principles that organize chromatin into these domains. And for that, Kai Huang, who was a previous postdoc in Yigal Schleifer's group, has developed the self-returning random walk, which is able to describe the statistical organization of chromatin structure. So starting from a specific point here, you can either jump forward with heterogeneous step sizes, or you can return back to the previous step. And this is what differentiates it from your traditional random walk. And it encompasses several important aspects of experimentally observed chromatin structure. First of all, this distribution of heterogeneous step sizes allows us to implement different epigenetic states. So for example, more euchromatic transcriptionally open regions can be better described by longer step sizes because each of the step sizes has the exact same number of nucleosomes, whereas these smaller step sizes result in more compacted chromatin. In addition, the self-returning property allows a more hierarchical organization which obeys the scaling laws that we are able to observe experimentally. So overall, the self-returning random walk forms this tree-like data structure where you have these tree domains which are coming out of this overall open backbone structure. And the overall statistics are only determined by this one folding parameter alpha, which determine the overall organization of the tree domains versus the backbone structure. And now that we had the self-returning random walk, one problem for using it to represent the chromatin polymer is that it disobeys two important components of polymer models. First of all, the self-returns result in a lot of overlap, so you can see each of these are different colored monomers that return to the same step, and in addition, it doesn't have this linear chain topology. So Marcelo Carignano, who's a current senior researcher in Eagle's group, developed an optimization procedure to turn this statistical model of chromatin into a self-returning excluded volume polymer model. And next, we wanted to investigate if the statistical chromatin structure represented by the self-returning excluded volume is still maintained upon different conditions. So we exposed cells to heat stress to induce a heat shock response in order to see if exogenous stressors caused rearrangements in statistical chromatin structure and if SREV statistics continued to be able to accurately describe the chromatin system. So first, we performed live cell partial wave spectroscopic microscopy experiments. So Vasundhara Agarwal performed these experiments where she had ACT116 colon cancer cells. We have a control population exposed to normal temperature, and then a heat shock population exposed to 42 degrees Celsius for 1 hour. And what we see here is a statistically significant increase in the average chromatin packing scaling. And what you can see in the actual packing scaling maps of these PWS experiments is that when you heat shock the control population, the green regions, which demonstrate low chromatin packing scaling, decrease, whereas the red regions, which demonstrate high chromatin packing scaling, increase. So next, we wanted to see if the self-returning excluded volume polymer, by only changing the one folding parameter alpha, would be able to accurately describe these observations. And what we can see here is that not only do we observe an increase in the packing scaling of this SREV model at the population level, but when we look at individual conformations generated by this SREV statistics, we also see a very wide heterogeneous distribution of the average packing scaling of each of these conformations that's representative of the distribution observed experimentally. So now that we've determined that packing scaling changes are caused by heat shock, we next wanted to assess how the overall genome connectivity, specifically the statistical genome connectivity, changes upon heat stress. So we performed high throughput chromosome conformation capture experiments, which demonstrate that at low length scales, the overall contacts between different chromatic regions actually decreases, whereas at longer length scales, the chromatin contacts increase. And by changing, again, just one folding parameter, we were able to recapitulate this statistical rearrangement of genome connectivity using the self-returning excluded volume model. So altogether, what we've been able to observe is that upon heat stress, the overall statistics of the SREV model is still or the overall structure can still be described by SREV statistics, but the folding parameter alpha is what is changed here. And when we visualize the one conformation of SREV for the control versus heat shock condition, we can really see stark differences in the overall statistics and connectivity of the chromatin polymer. So you can see the backbone in red, that there is much more backbone structures with smaller domains, which can be observed by the gray circles here. And these domains are much more connected in the control conditions, whereas in the heat shock conditions, the domains are larger and they're more interconnected, which increases the long-range contacts that are observed, whereas we're decreasing the short-range contacts. So overall, we have seen that the self-returning excluded volume model is able to kind of accurately represent changes in chromatin structure with just one returning parameter. So overall, what we've seen here is that chromatin is organized into these higher order spatially separable packing domains that are defined by chromatin packing scaling behavior, that the packing domains themselves have heterogeneous structural properties, and that chromatin organizes into domain-like data structures describable by the SREV model with statistical rearrangements upon exogenous heat stress. So now that we've been able to identify the existence of these structures, we next wanted to study their functionality. So what we see here is that chromatin state can actually influence what's called the phenotypic plasticity of cancer cells. And what I mean by that is in the normal conditions, there might be certain cell state transitions that are allowed, whereas maybe certain energy barriers might not allow cell state transitions under normal conditions, but lesions in supranucleosomal chromatin structure might decrease these barriers and allow for cell state transitions into a more malignant, potentially chemoevasive state. So we wanted to answer or figure out maybe how chromatin packing might be influencing specifically transcription, which might affect the phenotypic plasticity of these cells. And in order to do that, we were able to model transcription in a realistic chromatin environment by accounting for the phenomenon of macromolecular crowding. So you can see, for example, this gene in blue, which might be transcribed by polymerases and transcription factors, and it's surrounded by these orange crowders. And what crowders are any macromolecule, so this includes chromatin density, mobile crowders within the nucleus, cholesterol, anything that excludes volume but does not participate in the chemical reaction in transcription of these genes directly. And what we see here is that actually a kinetic model of transcription developed by Matsuda, who was a postdoc in Yigal Schleifer's group, is that crowding can have a very large effect on gene expression. And when we account for it, we see that there's this complex non-monotonic behavior where at smaller crowding fractions, an increase in binding of transcription factors and polymerases and their efficiency of binding actually increases the gene expression up to a certain point, where the decrease in diffusion of these reactants actually decreases this gene expression. So because chromatin density is the largest crowder within the nucleus, we next wanted to establish exactly how the statistical distribution of chromatin density might influence large-scale transcription patterns. And in order to do this, we have developed this chromatin packing macromolecular crowding model, and this was work that was begun by Wenli Wu and Luaya Almasalha. And basically, this model doesn't just consider molecular regulators of transcription, and these include concentration of transcriptional reactants such as polymerases, but also physical regulators, which define the statistical distribution of chromatin density within packing domains. And these three physical regulators of transcription are the average chromatin density within the domain, the genomic size of the domain, and the domain packing scaling. And we've seen that actually all of these physical regulators contribute to transcriptional responsiveness of genes to external stimulation and have specifically focused on the phenotypic or contribution to phenotypic plasticity of the domain packing scaling. So there are two distinct aspects of phenotypic plasticity that can allow for cancer cells to be, for example, more chemoevasive and more malignant. And one of these aspects that we consider explicitly is transcriptional malleability. And this is because cancer cells that are able to rapidly alter their gene expression to overcome cytotoxic stressors might be able to evade chemotherapy better. And this is changes in the mean levels of gene transcripts. And what we're modeling here is that cell populations that have packing domains with a lower packing scaling are compared to cell populations with a higher packing scaling. We expose both cell populations to the same cytotoxic stressors, such as chemotherapy, and then look at their average upregulation of these transcripts. And looking here at the overall upregulation of the transcripts in response to chemotherapy, you can see that the enhancement due to the higher packing scaling of the high D populations is increased for genes that are the most upregulated. So this means, for example, that the most upregulated genes of the high D population are 4-fold more upregulated than the low D population. And this is especially true for genes that have an initially low pre-stimulated expression, which might have important consequences, for example, for stress response genes that have low expression at baseline, but might need to be upregulated at a very fast rate in response to cytotoxic stress in order to evade chemotherapy. So in addition, besides encompassing the malleability of transcription, we also wanted to encompass the overall heterogeneity. Because we've seen that in cell populations that are exposed to chemotherapy, there might be a role in terms of the intercellular heterogeneity or the number of functional states that the cell population has access to in determining their responsiveness to chemotherapy. So again, we see this very strong match between single cell sequencing experiments and our CPMC modeling. On the x-axis, we have the chromatin packing scaling of populations. On the y-axis, we see there's an overall positive correlation between the packing scaling and the heterogeneity in our cell populations. So now that we have really been able to draw a relationship between the packing of, or the chromatin packing state of cell populations and their transcriptional responsiveness to chemotherapy, we next wanted to draw the line even further to determine cell phenotype. And this is work that I've done with Jane Frederick. And basically what we're considering is that a cell makes a survival decision at a critical time point after cytotoxic exposure to chemotherapy. And at this critical time point, if certain genes that are critical for cell survival are upregulated beyond a certain threshold, then the cell is much more likely to survive and evade chemotherapy rather than succumb to cell death pathways. And just like a side note here, my mom is from Greece, so as any math equation needs a certain Greek symbol in order to be able to characterize this, we've decided that theta is the Greek symbol here because in Greek, thanatos means death. So it very much fits well with this cell death pathway. So basically, we see again from our previous studies that there's an increase in both the malleability and the heterogeneity for high D compared to low D populations, and this contributes directly to the overall survival of this cell. So importantly, we performed experiments on the same dishes in response to the same exact chemotherapeutic stressors, and what we see here that's we are able to recapitulate with the model is that the lower D cells actually have an increased death probability, whereas we increase the packing scaling of cell populations, there you see this overall sigmoidal decrease that both the model and experiments show. So now that we've been able to kind of see the influence of initial chromatin packing state at the level of individual cell populations, we next wanted to test a theory that if we have a cell population that has an initially higher packing scaling, we expose it to some sort of drug agent that lowers the packing scaling, and then we expose both of these populations to the same cytotoxic stressor, then this chromatin-dependent adaptability model that we've developed predicts that the higher D population has more cells beyond the survival threshold, so decreasing the D of the population will actually increase cell death and therefore increase the efficacy of chemotherapy. And what we see is just that. So from the modeling and experiments, what we did experimentally was we exposed cell populations to different concentrations of chemotherapy to induce different amounts of cell death for the high D population. Before chemotherapeutic exposure, we lowered the D of this high D population, and then we see actually that exposing the different populations to the same amount of chemotherapeutic stress results in this exponential increase in cell death for this low D population. And we're able to see this both experimentally as well as with the model, which is able to explain basically mechanistically that lowering packing scaling will increase the cell death of a cytotoxic stressor such as chemotherapy. So overall, this kind of has implications for potential drug screening in the future. If we're able to find drug agents that are able to change the statistical packing of chromatin, then we might also be able to predict its response to chemotherapeutic stressors. So in summary, we were able first to identify the existence of these packing domains, which have these heterogeneous properties. And then with our modeling, we were able to also determine that the average size, density, and packing scaling of average properties of cell population will influence their transcriptional responsiveness. And specifically that the initial chromatin packing state and the chromatin packing scaling will modulate the phenotypic plasticity and cell survival to external stressors. So now that we have kind of related a very important aspect of chromatin structure to chromatin function, we next wanted to investigate another realistic part of the nuclear environment that we think might be able to strongly influence the statistical chromatin structure. So again, we know that chromatin is a highly charged polymer due to the phosphates of the DNA within the chromatin. So we really have this underlying question: how does the internuclear environment, including ions, pH, and chromatin density, influence the structure and charge of DNA and chromatin? And previous studies that have tried to answer this question have not been able to characterize this fully due to intrinsic limitations to these studies. For example, experimentally, we have single molecule studies that are able to observe the compaction of chromatin arrays, but these are not observed in physiologically dense systems. Additionally, we have cell studies where we can modulate the intranuclear environment, but we can't control other factors that might be influenced by changes in, for example, ion concentration, such as changes in enzymatic activity. In addition, computationally, we have very similar limitations. For atomistic simulations, which explicitly consider all of the interactions that are occurring at an atomistic detail in your system, this is able to represent your system with a high level of detail, but it's very difficult to perform these simulations for larger and denser systems computationally. In addition, we have these less detailed, more coarse-grained simulations, which are able to potentially be able to model these denser systems, but they represent interactions, and specifically electrostatics, less accurately to a level that might influence the overall results. So what we've used here in this study is molecular theory, and this is a theoretical approach that was actually developed originally by Egal Schleifer to investigate polyelectrolyte layers. And here we're using this approach to look at two different systems. The first is a simpler DNA phosphate loop system. So we're specifically here modeling phosphates, which are the only chargeable units of DNA, and we're looking at loops because in the nucleus there are a lot of topological constraints of the chromatin that will cause the chromatin to form these more elastic loop structures. And then we've built off of this DNA phosphate loop system to be able to study the chromatin system as a whole. And this is research that was performed with research assistant professor Rickert Knapp, who helped a lot with developing the theoretical approach as well as the software. And just quickly going over the molecular theory benefits, it explicitly considers the size, shape, charges, and chemical state of all molecular species, and thus links the structural state with the chemical or charge state, so giving us a lot of accuracy in terms of our calculations. And additionally, it's computationally feasible to perform parameter scans over a range of environmental conditions, including for dense systems. So this approach involves inputting system parameters such as concentrations of ions, pH, and density of the system. In addition, the charge and size of all molecular species, the free energy of all chemical reactions that are explicitly considered, as well as a representative set of chains. And this representative set of chains is very important because what the molecular theory does is that it determines the probability distribution of all input conformations. So for example, you can see on the bottom here, we've generated a distribution of DNA loop chains by performing molecular dynamic simulations, and we've performed them so that we have a range from more extended to more collapsed conformations. And we input this into the theory, which is defined by a system free energy functional that takes into account the different aspects of the system, including the conformational entropy of the polymer, as well as the translational or mixing entropy of the solvent, the chemical potential of the system, the electrostatic potential, as well as steric repulsions, which allow for excluded volume interactions. We turn this free energy functional into a system of nonlinear coupled integral-differential equations, and then we numerically solve this in order to output any thermodynamic or structural quantity of interest, and this includes the system free energy, and specifically I would like to point out that we're inputting the bulk ions, pH, and density, but what we're outputting is the actual three-dimensional distribution of these properties. For example, you can see the three-dimensional polymer density distribution on the bottom, as well as other properties. So turning to the simpler DNA phosphate loop system, we explicitly incorporate charge regulations via chemical reactions. And this includes the acid-base equilibrium, so the protonation-deprotonation reaction. Because phosphate is such a strong acid, the pKa of phosphate is actually 1, which means that at pH of 1 under ideal conditions, half of these phosphates are going to be in their negatively charged state. And this means that at physiological pH, which is about 7, that most of these phosphates will be in this negatively charged state unless there's some sort of other chemical reaction that neutralizes this charge. And for these positively charged cations in our system, they can actually help to neutralize the electrostatic potential through two different mechanisms. One is ion confinement, where these ions can be localized close to this DNA loop layer, but not physically bound to the phosphates. And the second is through this ion condensation reaction between the positively charged cation and the negatively charged phosphate. And explicitly, we consider the binding of two physiologically most prevalent monovalent cations in the cell, sodium and potassium. In addition, we consider magnesium, which is the most prevalent multivalent cation, and this can occur through both one-to-one binding, as well as this ion bridging reaction that occurs between two phosphates and one magnesium to form bridges, as you can see here, either within the same loop or between loops. So first, we wanted to investigate how monovalent cations influence the structure and charge of the system, and how the bulk system density might influence this responsiveness to environmental conditions. So in order to change the bulk density, we modulate the distance between DNA loops, which is defined by this parameter sigma, where a lower sigma actually means a larger separation between DNA loops and a lower bulk density. You can see here that the overall three-dimensional distribution of the DNA phosphate density is pretty different for lower versus higher bulk densities. And we can also see that structural parameters, such as the average height, which is the distance between the engrafted polymer and the top of the polymer loop layer, is also very different between these two different conditions. So interestingly, we actually see that for both systems, after about 100 millimolars of potassium concentration, which represents very close to physiological monovalent salt concentrations, that the height is pretty sensitive to changes in potassium concentration. And we see that the higher density system has a larger height in general due to the negative or the electrostatic repulsions between neighboring chains, which causes chain stretching and an increase in height. In addition, this higher density system is less sensitive to changes in potassium concentration. And this is even more prominent when we look at the charge state of the system. So these are the average charge fractions of phosphates on the y-axis, and basically, if we don't reactions, this would be one at physiological pH. And it's really the ion condensation of the potassium that's causing a lowered charged phosphate fraction. And you can see that the higher density system has even more ion binding due to the high electrostatic charge density within this denser system, and that this is even less sensitive to changes in potassium concentration. So now that we've looked at changes in structure and charge of this system in response to changes in the electrolyte environment of monovalent cations, we next wanted to investigate this effect for divalent cation magnesium. And this is because of this interesting ion bridging reaction, which you can see can potentially perturb the structure, but also two phosphates are neutralized for the price of one magnesium. So we can see here that actually the range of height corresponding to even lower ranges of magnesium concentrations is increased by about 3 times compared to just our monovalent salt condition. We can also see that magnesium actually causes collapse of this DNA loop layer, meaning that after a certain magnesium concentration, the height is insensitive to increased magnesium. And importantly, we see that the free energy of the ion bridging reaction is strongly determining the sensitivity of the height to changes in magnesium. And specifically, that higher ion bridging results in collapse at a lower magnesium concentration. Now, experimentally, it's very difficult to determine what the exact free energies of binding are. So what we actually did was we performed this parameter scan, and then we took experimental observations into account that double-stranded DNA attraction and compaction of chromatin occur within a millimolar range of magnesium, and this allowed us to determine a physiological ion bridging coefficient. Next, now that we see that the structure is much more influenced by divalent cations than monovalent cations, we wanted to see these effects on charge neutralization. And if we look here at the electrostatic potential, we can see that the no magnesium condition has a much lower electrostatic potential, which is centered around the negatively charged DNA phosphate loops. Whereas when we add just 2 millimolars of magnesium, this electrostatic potential is significantly neutralized. In addition, we can see a very heterogeneous distribution of the charge state, which is represented by the electrostatic potential. And this also is represented by a heterogeneous distribution of local pH. So again, pH is negative log of the hydrogen concentration. So the local hydrogen concentration is going to determine the overall pH in the system. And you can see a very large difference in the average pH as well as the spread in pH for the no magnesium and just the 2 millimolar magnesium condition. And I would just like to point out here that for more coarse-grained simulations that are able to represent these more denser systems, they actually will most likely be fixing the charge and the pH of the system. And we can see here that it's very important to consider these lateral fluctuations in our system. So now that we have been able to kind of more thoroughly examine this simpler phosphate loop system, we next wanted to extend this to look at the effects of the physicochemical environment on chromatin. And this is an ongoing collaboration that we have with Juan de Pablo's group at University of Chicago, and I've been working very closely with Ranya, his graduate student. And basically, we have 2 different systems that we're studying for this chromatin system. The first is the single nucleosome composed of 147 base pairs wrapped around the histone octamer. And the second is this 8-mer nucleosome array composed of 8 nucleosomes, each connected by linker DNA. And basically, we represent each of these systems at molecular detail. So the histones are represented by their constituent amino acids, and each DNA base pair is represented by 1 nucleobase, 1 sugar, and 1 phosphate. And in order to take into account all of the chemical reactions in the chromatin system, we not only include the chemical reactions of the phosphates, so the acid-base equilibrium and ion condensation of these phosphates, but also the acid-base equilibrium of the amino acids, specifically the acidic and the basic amino acids here. And you'll see that taking into account the chargeable behavior of the amino acids really influences the chromatin system and makes it distinct from the simpler DNA loop system. So first of all, what you can see here is that the electrostatic potential distribution in 3-dimensional space is very heterogeneous. you can see the negatively charged DNA loops wrapped around the histones, represented by this more negative electrostatic potential, and the positively charged basic amino acid residues are in this green region here. And first of all, what we see is that the charge per nucleosome is slightly responsive to changes in bulk pH and fluctuations potentially around physiological conditions. And even though this doesn't look like large changes in the charge state, compared to changes in the DNA phosphate state within this regime, you wouldn't see anything, because this change here is due to the chargeable behavior of the amino acids. And another interesting aspect is taking into account where we performed calculations where we didn't account for ion condensation, which you can see by the solid line, and then we performed calculations where we did account for ion condensation of physiological monovalent cations, and you can see that the ion condensation has a very large charge neutralization effect on our system. In addition, we see that the average charge per nucleosome is much lower for the 8-mer array compared to the single nucleosome, and this is because the 8-mer array takes into account the linker DNA that connects the nucleosomes, and the negative charge of the linker DNA is very much contributing to this decrease in electrostatic potential, or the charge of the system. So now that we have investigated this, we next wanted to look at the effects of structure, or the effects of the monovalent cation environment on structure and charge of our 8-mer nucleosome chains. So the first parameter that we looked at is the radius of gyration, which is a polymer physics parameter that determines or really tells us the overall, hello?
I think that was
The overall compaction of the nucleosome array.
And you're supposed to come when?
Yeah, okay. Sorry, how do I?
You go to participants
Yes,
at the
for
bottom.
what time?
I'm so sorry.
I'll call her, Ranya.
Okay.
Yes, that's fine. Sorry, I have to cut you up because I'm watching the defense of my granddaughter.
Sorry, how do
Okay, I see you then tomorrow at 2:15.
I think it has to be Vadim because he's a
Vadim has
host.
to do it.
Oh,
He's the
okay.
host.
Vadim, sorry, could you mute Rana, please? Okay, I think she's fine now. Okay.
Γεια σου, Αντινάκη.
I'm very sorry.
Εγώ θέλω να την ακούω.
Yeah, είσαι στο Zoom.
Μα το είχα πατήσει γιατί πρώτα μου είπε να πατήσω το start the video.
Have Vadim make you the host, and then you could turn it off.
Because I don't think I'm the host.
Yeah, it says
Αν
you're
δεν
the host.
είναι καθόλου, αλλά έχω αυτή τη φυσιοθέραπη που έχουμε πει για πρώτη φορά. Αλλιώς δεν μιλάω καθόλου. Αλλά ευχαριστώ πάρα πολύ για το link.
Yeah, you need to
Ναι,
mute
ναι,
yourself.
γεια σου, γεια σου.
It says I'm not talking at all, and she keeps talking.
Karen, stop.
I'm sorry.
Okay,
I'm
I
sorry.
think that we're okay.
No.
Okay, sorry about that. Okay, we're just gonna continue until the
Try to
end.
mute everybody, you should, or
I thought everyone was muted. I think it's okay now. Okay, I'm so sorry about that. Okay, so basically, we're looking at the radius of gyration in response to changes in potassium concentration at 2 different system densities. So we have our lower density system, which is still in a dense environment, but less dense than our higher density system. And you can see that our higher density system has a lower radius of gyration corresponding to more compact conformations. And this is because of the surrounding chromatin chains that contribute osmotic pressure and cause compaction of these chromosome chains. And you can see that our lower density system is more expanded and has a larger radius of gyration, and that this structural parameter is more sensitive to changes in potassium, but not so much above physiological monovalent salt concentrations. So now looking at the actual charge state of the system and its responsiveness to the electrolyte environment, we see first of all, that unlike the structural parameter, the charge state is much more sensitive to potassium beyond monovalent salt concentrations. So you can see that the charge regulation that's occurring in this regime actually isn't really influencing the overall structure of the system. And secondly, I just want to point out that even though it seems that the charge state of the two system densities is the same here, so you can see that the higher density system in orange, the lower density system in blue, that actually the charge density of the higher density system is going to be even greater because it's more compacted and overall denser. So finally, we wanted to look at these effects for local chromatin structure. So we see, first of all, these average changes in local structure, the nucleosome spacing between the neighboring nucleosomes, as well as the bond angle between neighboring trinucleosomes. And we can see here, I think most interesting thing is that the higher density system is more sensitive to changes in potassium concentration for these more local structural descriptors. So you can see the average nucleosome spacing decreases with increasing potassium concentration, but still this is even more pronounced for the lower density system. And this is true for the average bond angle. So you can see here actually, if you kind of think about it, is an increased bond angle means that the linker DNA, which is strongly negatively charged, is further away from each other in three-dimensional space. So this is why the higher density system has a higher bond angle, even though the average nucleosome spacing is decreased due to the osmotic pressure from surrounding chromatin chains. So overall, we have seen that even though beyond physiological monovalent salt concentrations, there isn't as much influence, the electrolyte environment still does influence the overall structure of chromatin for monovalent conditions. So overall, the summary of this molecular theory work is that molecular level interactions depend on the physicochemical environment and determine the structure and charge of both DNA and chromatin. The second is that bulk system density, pH, and the electrolyte environment will determine the chemical or charge state and the structure of DNA and chromatin with local fluctuations. And then finally, and this is kind of a future direction for the chromatin system, is that divalent cations have a much larger effect on the charge regulation and the structure of these DNA phosphate loops, and we anticipate that this will have a larger effect on the chromatin system as well. So with that, I just kind of want to briefly summarize the major conclusions of my thesis work, which was performed in collaboration with many different people that have been named here. So first of all, chromatin organizes into spatially separable packing domains with heterogeneous and responsive statistical properties. The second is that the statistical packing behavior of these domains actually influences their transcriptional responsiveness and will influence their adaptability and plasticity to external stressors. And then the third is that we really need to be considering all aspects of the physicochemical environment. So this includes the ions, pH, and bulk chromatin density in order to both regulate the charge and the structure of chromatin. And with that, I would like to thank, first of all, my research group, predominantly Vadim and Igal. Thank you so much to both of you for your incredible mentorship over the years. Like, I've really grown as a scientist because of everything that you have kind of taught me, as well as all present and past members of the Bachman and the Schleifer group, most of which who are named. I would also like to thank my thesis committee, Professor G, Professor Wu, and Professor Zadowska for their feedback and positive influence on my thesis work, and our collaborators and funding sources, and just kind of a fun acknowledgement here. I would also like to thank all of the friends that I have made in Chicago over the years, and here's some more. And also, thank you so much to my family, who's also been a great support to me during my PhD. And with that, I will take any questions from the public. Okay, any audience questions?
Anybody from the audience has a chance to ask a question. If anybody from the thesis committee wants to ask a question, it's a free, it's a free for all as well.
We have-
We will have a special time when we ask questions.
We have one audience question.
So Ranya, at the beginning of your PhD, you used to like do a lot of microscope stuff. Like you'd have to go in at like 9 p.m. and on the weekend and stuff. So where did that fit in here?
That actually
it's a lot of time.
fit into the ion work at the end. So like when I first started my PhD, I was doing experiments where I isolated nuclei and then I exposed them to different concentrations of ions. And I saw that the chromatin packing actually did change when I had the isolated nuclei under different ionic conditions. I think that the worry, I guess, is that it's not representing actual chromatin within the system, but I think that it still definitely did influence the trajectory in terms of our choices to pursue this modeling of that particular question.
I have a sort of similar question. So I know that a lot of the experiments that you did were sort of simulation experiments that you conducted on the cluster, but I know that also you did some more hands-on microscopy-based experiments. Yeah. I was wondering sort of what was the distribution between computational experiments and then more physical experiments and how that's going to carry into your postdoc work.
Sure. So I, honestly, during COVID, have not really touched a pipette. I have not touched a pipette. I think I started off doing the microscopy experiments. I think that it really helped me because I've been on kind of the borderline between the physics-based computational modeling and the experimentalists. And I think that it really helps me in order to model systems to be able to understand them and also the kind of maybe pros and cons of certain experimental methods and the types of analyses that we need to do in order to be able to answer questions about, for example, is the model valid, or what more experimental information do we need in order to inform this certain model. So I think that doing the experiments really helped me better understand the systems themselves. We have one more question. Oh, sorry. Okay, we can have as many questions as we want. Go ahead.
Okay, Sebastian asked one, so I had to ask one. Keeping in mind that I don't know anything about science and I don't know what a nucleus is, so when you talk about simulation stuff and you were simulating the loops,
yeah,
I'm assuming some of them are the probability distribution varies, so some of the structures are occur more rarely than others. Is that super expensive? So you have to simulate millions to get a few representative ones?
That is a great question. I thought about that. So actually, we have tried several different methods because, like you said, we do need a representative set of chains. One of the things that I tried to do was to change the solvent quality of the system in order to first, compact and extend certain chains. But it's really difficult to use different populations that are simulated with different conditions in this specific theoretical approach to account for those biases. So that didn't work out so well. So then actually Marcelo Carignano, who's in Igal's lab, helped us perform these molecular dynamic simulations where we actually had to simulate with a bias potential. So we have a 2-well bias potential for the free energy of the system that will bias towards the more compact and the more extended conformations, which are going to be very, very rare in your system otherwise. And then we subtract out the bias during the calculations.
Oh, that's swanky. So it's important sampling, kind of, but that's cool.
Yes. Thank you. Any other audience questions?
There's one question from the Zoom audience, Suri.
Hi, how are you? It's very nice talk. I'm so from Professor Zdowska lab. I want to ask about the kind of distribution of this packing domains. Yes. Because you measure the size and then the kind of mass scaling curve within a domain. But how this mass scaling changes, kind of when you extend your graph outside of domain area, and what's the distance between domains, because that's also directly correlated to the contact probability in the high CMAP. And you show the certain change in the contact probability in the high CMAP. Does it appear in your packing domain distribution change or something like that?
Yes. So actually, I'll show you 2 different things. The first one is we've performed analyses. So the ones that I showed you were looking at the single domain regime, but these are ensemble results. So starting at randomly selected points within the nucleus, and then looking at this overall distribution. So we see that there is like this packing scaling regime, and there's one packing scaling coefficient that's able to define this regime, and then it slowly actually increases up to a d equals 3, indicating some sort of random behavior or random distribution. So that's what it looks like on the population level. So I think it is kind of similar to the high C in terms of there's kind of like a shorter range versus longer range behavior. And I think like first of all, like the d equals 3 kind of shows that there isn't some sort of higher order supranucleosomal structure. But I think it's interesting that there's kind of a continuous change of the scaling. And I'm wondering if that also might be due to the fact that we're looking at this in individual nuclei instead of aggregates over millions of cells.
One small follow-up kind of question. You calculate the mass scaling curve from your like TEM image. So, I mean, is that possible because like you had a tomography of it? Because like normal 2D TEM image probably is not reliable to like directly calculate the mass scaling curve out of it, right?
So kind of two responses to that question. The first is that with Chromestone, we can get a 3D-dimensional representation of the tomogram. So this allows us to get mass scaling information up to about like 100 or several hundred nanometers, or like 100 nanometers, which is the size of the section. So this kind of gives us validation that there's this packing domain region, which has like this specific packing scaling behavior on aggregate. And then we actually, we did perform 2-dimensional mass scaling, where we project into this one plane and then perform mass scaling. And we see a very similar regime in terms of the extent of the regime. You can see the packing, the fractal MS is the packing domain MS, and the supradomain MS is in red. And you can see that there's kind of very similar regimes for the 3-dimensional and 2-dimensional. There also is like a fractal coadditivity law that allows you to really compute fractal dimensions between dimensions. And we've also performed polymer simulations, and we've kind of done these projections for 2D and 3D in order to kind of validate that that's you know, realistic in terms of our system.
Okay, thank you.
Yeah, thanks for your question.
So I've been monitoring the chat, and there are a lot of comments. Everybody congratulates you with a wonderful talk. So there are a lot of very congratulatory comments in the chat, but no questions really. And maybe it looks like we don't have further questions, so maybe I just make one last comment on behalf of both Igal and me, and I think Igal will agree with that, that as Ranya's advisors, what we noticed is that Ranya is really an independent investigator nowadays, and has been an independent investigator for several years. So our job as advisors has been actually very easy. Just primarily listen to what Ranya has to say, and say this is really interesting. Research rules.
This is true.
And just not you know, keep nodding our head. And she has been a truly independent investigator, creator of ideas, and innovator for several years now. Any further questions? Maybe it's a good time to ask everybody who is not part of the physics committee to leave either the real